Hello, today we are going to talk about genetic engineering and how it helps us to treat the diseases like diabetes. There are two types of diabetes. The process we are going to talk about and discuss today is important for the two types. But we are going to talk about type 1 diabetes, which is sometimes turned to called childhood diabetes. In childhood diabetes, something goes wrong in the immune system. Our immune systems act as a police to protect our body when a virus comes in our body, but in type 1 diabetes, the immune system attacks the wrong ones. In type 1 diabetes, the immune system attacks the cells of pancreas, which are very important to produce insulin, which comes from Langerhans island cells. When diabetic patient body stop to produce insulin, they become very sick. If they did not receive the medication, they could die. So doctors give patients insulin to inject themselves so they can live very normal life. And that's what we are trying to do using genetic engineering. But not only injections, they sometimes get oral tablets to produce or help the pancreas to produce more insulin. First, they began to get insulin from cows or other farm animals to be used, but it was very hard because animals at first must be killed and pancreas removed. Scientists get small amount of insulin, so new techniques are required to produce huge amount of insulin. One of them, genetic engineering, provides us one. This new technique involves some bacteria, DNA plasmids, and remarkable properties of genetic code, which are we going to know it later. We all have DNA in our cells. DNA is a double helix. It's shaped like a ladder, but it is twisted around itself and they store information inside it. Each row contains letters as a code. They must be known how to read. Genetic code, it's divided into segments, each one called gene. Each give tells us how to make proteins. There are many types of proteins. Protein make our muscles. Protein produce pigment of our skin, hair and eyes. Protein also digests our foods. And it helps us to hear and talk to each other, and it helps our cells. Insulin is considered as a protein to do their function. The genetic code are like books, and there are a lot of books you can read, and only if you know how to read it. They can read any type of code that we give it to it, which means the cell. Now we are going to discuss genetic engineering. Genetic engineering is the insertion of the required code into the cell of an organism to produce a desired result. Like for example, what I mean is the insulin. The universality of genetic code makes the genetic engineering possible because it means if we take the gene of the insulin and place it into the cell of the other organism, then that organism can make the insulin that we are required to do. This process requires genetic engineering to cut the DNA at specific places. To do this, they use molecules called restriction enzymes. They act as a molecular scissors by using the right restriction enzyme genetic code. Genetic engineering can cut a DNA molecule wherever they want. To replace our favorite gene, genetic engineering uses collagenase molecule to paste the pieces of DNA together. By all that, we can produce human insulin, better than that produced from animals. Genetic engineering choose a type of bacteria called Escherichia E. coli. They used E. coli due to many reasons. First, because it is easy to grow this bacteria, and the other reason is simplicity. and it is cheap. Many bacteria contain very small pieces of DNA called plasmids. These plasmids are very useful because their small size makes them simple
even smaller
and easy to
pieces
work
of DNA
with,
called
and even
plasmids.
cheap
These
if you want to
plasmids are
reproduce
very
it again.
useful because
When
their small
genetic
size makes them
engineers
simple
need to place
and easy
a
to work
gene
with. So when a
into
genetic
bacteria,
engineer
they
wants to
usually
place
use
a gene into
to
a
add it to the
bacterium,
plasmid
they
and then
usually
replace
add
the
it
plasmid
to a
into
plasmid
the
first,
bacteria
and then
to
place the
let it
plasmid
reproduce
into the
once more
bacterium.
again. After a while, we found that the DNA of the bacteria changes and it starts to produce the required plasmid that it given inside it. And by that simple technique, we get the DNA that we required, that which means the insulin.